providing workshops on microbiomes eventually.
Okay.
And we ask a lot of questions as you go through.
Okay. Well, then I hope I don't go too far over time if you have lots of questions, but I'll do my best. All right. It's a pleasure to be here. Thank you very much for inviting me today. Let's see, I gotta find my mouse here. All right, arrow keys work. All right, so today I'd like to talk to you about permafrost on Earth, which of course includes many microorganisms. Relate that permafrost on Earth to permafrost and frozen ice on, or of course ice is frozen, sorry, ice on Mars, and to issues of planetary protection. Now, if you're unfamiliar with planetary protection, you might get a picture of these guys in your head when I say those two words. And indeed, part of planetary protection is protecting the Earth from any potential harmful impact of potential extraterrestrial organisms, okay? But the other half of it is protecting the other planets and bodies in our solar system from Earth microorganisms. Well, from Earth organisms, and for the most part, this has focused on microorganisms because of their hardiness and their unique abilities. So most planetary protection protocols that exist today are about protecting other planetary bodies from our own microorganisms. And I'll talk more about that as we go through, but just for a little introduction there on planetary protection. So let me begin by giving you a little introduction on terrestrial permafrost. Permafrost is soil that remains continuously frozen and has remained frozen for some time then. You can see in this graph that this is depth into the soil versus temperature. And you can see that the top portion is actually not considered permafrost because it does thaw out in the summer in some locations, not everywhere, and then freezes during the winter months. And this line is of average maximum and average minimum temperatures in these soils. This is a typical temperature profile from permafrost from the Arctic. It's get a little colder in the Antarctic, so the profile is a bit different. But that top layer that freezes and thaws is referred to as the active layer. And that freeze-thaw does give you this typical pattern ground that you see in this aerial view of some permafrost in Alaska. As you go deeper into the soil profile, the temperature does stabilize and then eventually rises above zero as the warmth from the Earth's core heats up the ground as well, counteracting all the cold surface temperatures. This other picture, the top picture, shows a cross-section through a somewhat thick layer of permafrost in Alaska. That cross-section is exposed by the river that flows through there. And you can see a little bit, some of those shinier looking areas are ice wedges that form within that permafrost as well. The thickness of permafrost does vary depending on location and what the average surface temperatures are in that location, and varies from tens of meters thick to the deepest is 1,500 meter thick permafrost that's found in Siberia. So to give you a better idea of where we find permafrost, it is mostly found in the northern hemisphere. All the purple areas are showing you different extents of permafrost, the darkest purple being continuous permafrost that covers all of the area in those places. The lighter purple is showing you where you have patchy areas of permafrost and they're not all connected to each other. Permafrost in the Arctic ranges from being organic rich to being organic poor. It just depends on where you are. And it varies by age as well. It didn't start getting cold in the Arctic until the end of the Pliocene, about 2.5 million years ago. So the oldest permafrost that we find is about 2.5 million years old, and that's found in Siberia, where I have the year mark on the map there. The oldest that we find in the North America is about 740,000 years old. There is also permafrost in Antarctica, not as much because most of Antarctica is covered by an ice sheet. Most of it in Antarctica is found on the Antarctic Peninsula here, where you have less ice sheet, and also in the dry valleys, which are right about here. Those areas are not covered by ice, so you can have some permafrost forming there as well. The age of permafrost in the Antarctic is less well constrained, but it's estimated that the oldest permafrost there might be 5 to 8 million years old. So when I think about microorganisms potentially living in permafrost, I want to put this picture in your head. These are frozen environments, and that freezing low temperature conditions really constrain what the microorganisms can do. This picture is actually from sea ice, but all we have to do is add soil particles into that. You'd have a picture of permafrost essentially. And what you can see is that as the temperature drops below freezing, ice crystals will begin to form, and as they form, the solutes don't get included into the ice crystal, so they're actually expelled into any remaining water that's present. And so this makes the liquid water that's present saltier, and that depresses the freezing point. So as you go with lower and lower temperatures, you end up with whatever liquid water is still there is very salty, okay? So it's that solid and salty nature of the liquid water that's still present in permafrost is an issue for the organisms living there. They're very physically constrained. They have to deal with those high solute conditions. And they as well, the bacteria get excluded from the ice crystals and are primarily found in the veins and pockets of brine that you can find in these frozen places. So along with the water getting stable, the macromolecules are getting stable too. So all of the biomolecules associated with the cell are becoming more frozen, more solid, and the organisms have to deal with that. As well, just because there's a reduced thermal energy because of the lower temperature, reaction rates decrease and diffusion rates decrease as well. Diffusion rates are doubly decreased, not exactly doubly, that's just a figure of speech there, but because of the solid matrix of the ice. Okay. So it's a highly constrained environment for these microorganisms. But despite those constraints, we do see that permafrost is in some sense alive. We know that microorganisms and other organisms can survive under these conditions for many, many years. For example, plants have actually been grown from seeds and fruits that had been frozen for 32,000 years within the permafrost. More recently, there was a study on reviving nematodes from 42,000-year-old permafrost. And we find that we can revive bacteria and other microorganisms from permafrost all the way back to ages of about 2.5 million years old. So we know that the organisms are able to survive. We also know that they're not just dormant while they're in this permafrost, but they actually have to do something to be able to survive, because actually long-term burial in that permafrost selects against endospores, which, as we know, have no metabolism going on. And in addition, studies looking at intact DNA in the permafrost were able to extract very large pieces of DNA from 400,000 to 600,000-year-old permafrost, which is also a sign that some kind of maintenance and repair has to happen to that DNA, because it's estimated that if DNA was just in this environment with no maintenance and repair, it would be sheared into very small pieces within 100,000 years under these conditions. So, okay. So there are lots of constraints, but we know that microorganisms can survive. And one of the big questions then that I'm working to address with my research in terms of this is, what adaptations have permafrost microorganisms evolved to live at these low temperatures? Then how are these adaptations relevant to ecological success and ecosystem processes? Now, this second question isn't particularly important, considering that with global warming, the Arctic is warming at a higher rate than the rest of the planet, and a lot of permafrost is thawing right now. And a lot of that organic rich permafrost in the Arctic is going to be above freezing now, and that's going to allow bacteria to be more active and potentially transform all that stored organic material into the atmosphere. So there's a lot of relevant work being done as well that my research is relevant to as well, but I'm not going to talk about that stuff today. I'm going to talk about planetary protection stuff. And for the planetary protection side of thing, one of my big interests is really understanding what limits the active reproduction of bacteria in permafrost. How do they survive that 2.5 million years in the permafrost? So to answer these questions, many approaches can and should be taken to address these. Today I'm going to describe some of the work that I and my colleagues have done to answer these questions. I'll talk about work I've done with model organisms in pure cultures from the permafrost, and then also talk about measuring activity at low temperatures, also both pure culture work as well as in situ analyses. Now, I'm not going to have time to get into all the details of this, but just to give you some idea that, you know, you got to throw lots of methods at this in order to really get lots of good solid data on what you're trying to demonstrate in these environments because of the restriction, the constraints on these microorganisms. I'm sure your pet microorganism has, you know, the same kinds of things, but just specific to your system. And all of the data that we gather from these kinds of studies is going to inform the limits of activity in these systems and then help inform planetary protection protocols as well. So I'm going to start with the pure culture work that I've done with this model organism, which is a species of Psychrobacter. Psychrobacter is a genus in the gamma proteobacteria. It's kind of your average run-of-the-mill gram-negative bacterium. Many species have been isolated. Most of them are isolated from cold and saline environments. The 26 species that are listed here, and then the habitat of their isolation is shown in the pie chart there, are particularly relevant to what I'm going to tell you today because all of these have genome sequences available, and I've done some comparative genomic analysis on this. Okay. So you can see that most of these come from marine environments. I did luckily have 6 genome sequences from terrestrial environments, and they were all frozen. So 4 came from permafrost and 2 came from soils that were more like the active layer, which don't necessarily stay frozen year-round. The 2 pure cultures that I've done the most work with are these 2 Psychrobacter, Psychrobacter arcticus and Psychrobacter cryohalolentis. They were both isolated from Siberian permafrost, and these 2 species, I think there's one other species, have been demonstrated by work I've done to grow in pure culture in the lab at -10 degrees Celsius. So I know they actively reproduce at -10 degrees Celsius. A couple of the other Psychrobacter in this genus, you know, again, only pure culture work will tell you this stuff, can grow at those very low temperatures. Not all of them do. Some of them cannot grow below freezing. Some of them can grow all the way up to 42 degrees Celsius. So we have a small range of growth temperatures within this genus as well, that some can grow down to -10, some can grow up to 42 degrees Celsius. Yep. At -10, it was around 30 days. Yeah, which is pretty good, I thought. It's a lot longer for other organisms. But I think that's because this is just like, you know, E. coli of the permafrost, this guy. So, all right. So the pure culture work that I've been involved with on Psychrobacter arcticus and cryohalolentis to look at what kind of adaptations microorganisms have evolved to live at low temperatures. Lots of different studies have gone into that, and we come up with 3 major themes on how you need to be cold adapted or how you can be cold adapted. Organisms need to maintain molecular motion, they need to stay in the liquid, and they need to conserve energy. And this is not something we see just in Psychrobacter. This is fairly common for most psychrophilic organisms that are living at low temperatures. And these bullet points here are just kind of a little more detail on how you achieve those 3 major themes of cold adaptation, and were nicely summarized in this recent review article on cold adaptation in psychrophiles. You don't have to read those right now. I'm going to go through them in the next couple slides with specificity to how Psychrobacter handles these issues. Okay. So Psychrobacter arcticus, we have the most data on, and for maintaining molecular motion, there are a number of changes in gene expression that occur during growth at low temperatures. And most of this are increasing the expression of genes that aid replication, transcription, and translation. You've got to have that stuff functional if you're going to be reproducing at these low temperatures. The yellow highlighted things on this slide are things that we see in Psychrobacter, but we don't see as commonly in other psychrophiles. So for example, here under gene expression, this protein CSPA, it's a cold shock protein, and it binds to single-stranded RNA to help keep that open and available for transcription and translation. And it is usually just induced at low temperatures, and many bacteria, E. coli has one too and does it for cold shock reasons. But in Psychrobacter, we see that it's constitutively expressed at all growth temperatures. And so that's something that's a little bit different there. Proteins need to remain flexible. We see amino acid changes in proteins that lower the thermostability and allow them to remain active and still have good activity at those low temperatures. I'm going to talk about that in a little more detail coming up. And as well as the isozymes there. They've got to maintain membrane fluidity, right? You probably heard this in Bio 110 if you were an undergrad here at Penn State, whatever your intro bio class was, right? So more unsaturated and short chain fatty acids in those membranes at low temperatures. We also see some cell wall changes. Again, not clear if this is unique, how unique this is to Psychrobacter, but it has not been documented in a lot of other psychrophiles. Of course, you never know if that's just because nobody's looking, right? Because not everybody looks for the same thing. And to combat those high solute conditions, they accumulate compatible solutes, as many organisms do to deal with higher salt conditions. We see that to help maintain functionality at low temperature, often psychrophiles have a redundancy of genes for these genes that are needed to help maintain processes within the cell. So you see their chaperones, that cold shock protein, multiple copies of transfer RNA, things like that. They seem to have more biogenesis, cell membrane biogenesis genes than you might find otherwise. And it's believed that some of this redundancy of genes is involved in something we call allele switching, where one copy of the gene is expressed at low temperatures and presumably has better activity at low temperatures, whereas the other copy is expressed at high temperatures and presumably has a better activity at that high temperatures. So they may be tailoring each of those alleles for working at specific temperatures. And we have some data to support that, but we need to do more to really understand if that's what's going on there. And conserving energy, this would seem logical. Most microorganisms have to go out there and conserve energy, right? But again, not very well documented in other psychrophiles. We do know that Psychrobacter have very efficient growth at low temperature. So when you compare the temperature at which their growth yield is at a maximum, it's about 10 to 15 degrees lower than the temperature at which their growth rate is at a maximum. So they're much more efficient at those low temperatures. That's also been documented in some sulfate-reducing bacteria as well. And when we look at gene expression, we can see that there are various energy-conserving pathways that are induced as well at the low temperatures. Okay. So I want to say a little more about amino acid composition changes within Psychrobacter. Again, this is something that's been studied for many years because it's always interesting to know how enzymes function at different temperature regimes. There's a lot of work done in thermophiles as well. And this is a summary of what is kind of known within the community about cold-adapted proteins. And in general, you see that they need to reduce stabilizing interactions so that they can maintain their flexibility at low temperatures, just as thermophilic proteins have more stabilizing interactions in order to stay together at the high temperatures that they work at. Again, this was all summarized nicely in this review article. I'm just going to point out a few things here that we're going to follow along in the next couple slides. I'm going to use proline and acidity as my examples here. So cold-adapted proteins are going to have fewer prolines, and they tend to have fewer acidic residues as well. Okay. So interestingly, psychrophiles show these changes in amino acid composition, and those psychrophiles that grow at the lowest temperatures, it's kind of easier to see these signatures of cold adaptation in their proteins. But it's not so easy to see them in Psychrobacter. And we think this might have to do with the fact that Psychrobacter is not your classic psychrophile as defined way back in the 70s by Professor Morita, that he put 20 degrees Celsius as the maximum growth temperature for a psychrophile. How he got 20 degrees, I don't think anybody really knows. But most Psychrobacter can actually grow above 20 degrees. And so they're not your classic psychrophiles in that sense, but they grow at -10. And their growth yield is certainly maximized below 20 degrees Celsius, not above 20 degrees Celsius. So by other definitions, I would certainly think they are considered psychrophiles. but it's not by that classic definition. And most of the work that's been done on cold adaptation have been done in these kind of classic psychrophiles that don't have maximum growth temperatures over 20 degrees Celsius. So it's not entirely clear why, but these cold adapted signatures are a little bit harder to see in Psychrobacter, but we still can see them. We have to use slightly different techniques. So here's our proline again, and what was done in this study is that a gene was compared to its homologs, and the proline composition was compared. If it was lower in the Psychrobacter, that was considered a cold adapted protein. If it was the same or about the same or higher than its relatives, or mesophiles, then it was considered not cold adapted. And we do see that when you go gene by gene through the genome of Psychrobacter, that more of the genes do show a cold adaptation than not. So we can still find it, but it's not, the signature is a little bit obscured. And you see that the genes that show most of this cold adaptation are, not too surprisingly, core housekeeping genes that we really need to keep functional at all the temperatures the organism is going to grow at. So replication, translation, amino acid, metabolism, everything like that. So we do find the genes that show these cold adaptations are in those core housekeeping functions as well. We can see it in those isozymes that do the allele switching as well. So here's just two graphs showing two examples of this. An RNA helicase where one allele is more expressed at the lower growth temperature and the other at the higher growth temperature. And when you look at the sequences of these, you can see some cold adaptation in the one compared to the other as well. But again, it's not always so straightforward to pick out, because any of these changes can lead to cold adaptation, any of those changes that we saw. And sometimes you only need one change in an amino acid to make something cold adaptation. So sometimes it is hard to pull out what change in the amino acid composition is leading to a cold adaptation. So this has been seen in Psychrobacter arcticus and Cryohalentis, as well as in this Rhodococcus species, which was isolated from Antarctic permafrost. Okay. So we see that overall, Psychrobacter kind of looks like other psychrophiles in terms of what it does for cold adaptation, but there are some differences. Okay, allele switching hasn't been documented that much. You know, the signature for amino acids is a little harder to pull out, and we're not sure why this is. We do know that most data comes from organisms, most data on cold adaptation comes from organisms isolated from marine environments, and they do have different constraints there. The marine environment tends to have more stable low temperature conditions, a higher salinity, and a higher diffusion rate because it tends to stay more liquid. Compared to terrestrial environments where we see fluctuating low temperatures, especially in that active layer, right? So most organisms in the permafrost actually have to pass through that active layer before they get buried into the permafrost. And when it's not frozen, there's going to be a much lower salinity in these terrestrial environments. When it is frozen, it should be about the same as the marine, but you're going to have lower diffusion as well because there's going to be less liquid water there and more soil particles and ice crystals as well. And this graph shows nicely, this pie chart is showing genomes of cold adapted bacteria that have been sequenced. And you can see that about half of them are from the marine environment, and there's just that tiny little sliver of organisms from ice or permafrost. So understanding the differences between frozen environments, marine and terrestrial, is a little more challenging just because of the paucity of genome sequences available at this point. So the genus Psychrobacter is actually a nice genus to look at this in because we do have isolates from the marine environment, from permafrost, and from some other places. And we had, at the point I did this study, we had 26 genomes. Of course, every day you look, there's more genomes out there. But at this point, we had 26 to work with. And I compared these 26 genomes of Psychrobacter to try to get some idea if there were any differences based on habitat between these organisms. They are fairly closely related as genera go, so the individual species are fairly closely related. So in terms of gene content, they share quite a lot of genes. So the average Psychrobacter genome is about 3.2 megabase pairs, has about 2,800 genes, and about 1,400 of those are shared among all the Psychrobacter species that were analyzed, which is fairly high. If you compare it to other genera, that is fairly high, shared within a genus. Those core genes are primarily found in where we would expect them in housekeeping functions. So the red bars here show the the core genes for the Psychrobacter species. Most of them here in translation, amino acid metabolism, energy conservation genes, coenzyme transport here, and membrane and wall biogenesis genes. So not too surprisingly, they share that suite of genes that allow them to do all the core housekeeping functions. The genes that are in the cloud on that graph are those genes that are unique to each genome. And not too surprisingly, those are mostly in the mobilomes. These are all your transposons and other kinds of mobile elements that you find in each genome. And then the shell genes are in between. They're not core genes. They're not unique to the genome. They're shared by 2 to 24 genomes. And there was just a variety of genes there as well. There weren't any super trends that we could see there in terms of content and organism. When we divided those up into 3 major habitat groups, there were Psychrobacter from marine environments, from the permafrost and frozen soils, are in the terrestrial group there. And there were actually 4 Psychrobacter that were associated with warm hosts. 2 Psychrobacter species isolated from humans, and 1 from a seal and 1 from a tropical fish. So these 4 Psychrobacter were a little bit different, and I put them in their own group for that reason. They also were some of the Psychrobacter that grew at the highest temperatures and were unable to grow at very low temperatures. So when we looked at kind of habitat-specific genes, nothing super straightforward for the terrestrial and the marine organisms. Terrestrial and marine, there was a lot of variability there. They certainly shared more genes with each other than they did with the host, but there were no genes that were specific just to the terrestrial genomes or to the marine genomes. There was just a lot of variability there. Interestingly enough, the host genomes, the Psychrobacter from warm hosts, did have 33 genes that were found in all of those genomes, and suggest that maybe there is a small suite of genes that are necessary for being associated with a warm host. So that was an interesting finding, not my major focus of my research, but something pretty interesting to follow up on as well. So unfortunately, no smoking gun here for genes needed to live in the permafrost environment, but that's not too surprising either. We can still see the changes in amino acid composition that look like cold adaptation. So this is a PCA plot, just looking at not all of the amino acid composition, but some of the ones that are most notable for cold adaptation, like proline is here. And you can see that the organisms group a little bit by habitat. So here's all our organisms that are associated with warm hosts, and not too surprisingly, they look a lot more mesophilic in their amino acid composition than the other Psychrobacter. Particularly, they have much higher proline content. All the marine and terrestrial Psychrobacter are over here. Interestingly enough, the terrestrials kind of move over here, and we can see, as was verified by some ANOVA analysis of these data, that they do have a significantly higher aliphatic content than the marine Psychrobacter. So the proteins from the terrestrial Psychrobacter appear to be more hydrophobic. So we see that in terms of aliphatic index and the grand average of hydropathy of the proteins. Hydrophobicity is known as something to change with cold adaptation, but it is one of those signals that's sometimes even hard to find in the classic psychrophiles, because often what happens is that the exterior of the protein becomes more hydrophobic and the interior becomes less hydrophobic with cold adaptation. So those 2 contrasting effects kind of cancel each other out when you're trying to look for a cold adapted signature in the amino acid composition. But for some reason, it seems that the terrestrial isolates do show more hydrophobicity than the marine isolates, and I don't know why yet. I don't understand what that would be for. But we can see that this graph is showing for the individual genes that and the cold adaptations they show, it does look like the marine isolates are, I can't really say preferentially, but proline seems to be more common cold adaptation
in the marine genes than in the terrestrial ones. Whereas here is the terrestrial, the hydrophobicity and allophobicity, where
the terrestrial organisms seem to have that more commonly as a change that results in cold adaptation. So, interestingly, they do have some difference in amino acid composition than the marine organisms. It's not much, but it's a little bit different. This might still be confounded by the number of genomes that I've analyzed. Of course, more genomes is always going to potentially change your conclusion, so I'll be interested to see how this changes as more genomes become available. Unfortunately, most of the genomes that become available are still marine organisms, so I have to make a big effort here to get some more permafrost genomes available for the Psychrobacter. So we'll see how this plays out. Okay, so that's kind of the end of my pure culture work that I wanted to talk about. And all of this really gives us the confidence that these bacteria have the potential to be active at these low temperatures within the permafrost. Okay, so now we're going to switch a little bit to think about, yep.
I just want to ask a quick question about what the dispersal in the permafrost environment is like. Like I would expect the ones living in a marine environment are all over the place, and in a permafrost, like I don't know what the liquid flow is like in these brine channels. Like it's something 58 generations removed from its ancestors, like in centimeters away from it, or has it been?
It's probably very, very slow. So they're probably not moving at all. And so, yeah, I don't think that the bacteria, I don't think it's very well known, but a lot of studies done in the soil science community have shown that like once you get to about -7, you can't really detect respiration anymore. And so that's kind of when activity really falls off. And it's because it's thought at that point that you are starting to even disconnect all these little liquid veins from each other, and you're really not getting much dispersal or diffusion through that.
And I wondered if you have like much stronger biogeographic patterns.
Yeah, so that's part of what I think might be going on with the gene content differences, is that you're going to have these isolates that are separated from each other in the permafrost. They've been separated for 20,000, 30,000 years, and so they're having some niche specialization and maybe interacting with a few neighbors, but not very many. Yeah.
So I have a question.
Yep.
Maybe you're about to get to this, but I'm curious if these signals that you have been detecting that are differences in cold adaptive alleles, if they, if those are signals that you might be able to pick up in many genomes.
It's a little harder, but I think it could be done. It's you gotta. I would like to have a bioinformaticist on my team, and yeah, do that work because it would be very interesting. But the potential's there. I've tried to do some of that, and it's just a little difficult. But the potential's there to do that. Yeah. All right. Good. Recruit some people here to help me out. All right. So we want to better understand how active those microorganisms can be in situ, considering that the amount of activity is going to depend on these many things: liquid water, adequate energy and nutrients, the absence of damaging conditions. This is of particular concern in permafrost because it is a soil environment. There are radioactive minerals here. And over that 100,000 year time frame, you can accumulate a bit of damage. The low temperatures, and then the time to evolve adaptations, because our planet actually hasn't been all that cold for all that long. Antarctica has been that cold for about 35 million years, okay? But before that, the whole planet was fairly warm. Okay? So it's not clear that any adaptations that had been evolved in previous cold periods would have survived to the current cold periods that we have on our planet. Okay, so here's my obligatory table you can't really read. And this table is a summary of the many studies that have been done in frozen environments, trying to understand how active organisms can be. And so people have looked at a variety of temperatures from -5 to -33, different cellular processes to document activity. Respiration is a common one to look at, protein synthesis as well. Cell division gets looked at, but it's often more challenging to look at as well. And I don't need to go too much into this, but I just want to highlight that a lot of these have been done with permafrost organisms and microcosms. So we are trying to get a handle on how this is in permafrost, not just in like sea ice or other frozen environments that we might have as well. Okay. So I'm going to summarize this in the next slide, where all of that data that has been accumulated from many studies, so far we have, in terms of limits on reproduction of bacteria, the lowest documented cell division has been at -15 and -18 for yeast. We can see protein synthesis at even lower temperatures, and then various types of respiration all the way down to around -30. So we know that there's some kind of metabolism going on and that it is possible. It gets hard to demonstrate these things because these experiments start to take years, and grad students don't want to stick around for that long generally, so you got to be careful with that. And most of those were done in kind of pure culture conditions in the lab, which are a lot more ideal, right? There's usually more nutrients there. They're not as realistic to what the organism would see in the environment. Microcosm studies have been done to try to get a little more realistic as to what might happen in the environment, and we see, you know, some of these same things occurring at the same temperatures. But again, it's just these take even longer than the pure culture studies generally, so there haven't been as many studies done that are documenting these or at quite the same very low temperatures either. Okay. All right. So these studies suggest, you know, these pure culture and these microcosm studies suggest that bacteria should be able to be active in the permafrost, but we can also go straight to the permafrost. Some of this work is from, because I think in Russia they don't care so much if you add radionucleotides to the permafrost. They have done a couple studies like that, but there's not too many studies quite like that. But we can go to the environment and just take other measurements to assess whether or not activity is occurring. And here I've got 3 case studies where we go to the environment itself, which has been under these low temperature conditions for a long time, to try to get a feel for whether or not microorganisms can handle these low temperature conditions. Okay, see I'm going over time. I'm gonna be a little long, so I'll try to get moving. So Arctic permafrost doesn't experience, actually at depth, the temperatures only get down to about -15, so it's not the coldest place to look at this, but it is, we know reliably that it's been that cold and frozen for that long, up to 2.5 million years. So we know that very well about the permafrost. The water there is in very thin films. As I had mentioned before, we can get viable and cultural microorganisms out of here. And we know, because endospores don't survive a long period of time, that some kind of maintenance activity has to go on, because we can get that large chunks of DNA out as well, that some kind of maintenance goes on, that these viable and culturable microorganisms that we pull out have to have done something to survive that 2.5 million years in the permafrost. So we know that even under those, you know, very thin films of water, -15 degrees, and low conductivity, that yes, some activity occurs. But it does seem to be very little activity, because we see very little degradation in situ of the soil organic matter. A lot of the permafrost in the Arctic is very rich in organics. This is a good example of some organic rich permafrost. You know, plants grow on the top, and then slowly they get buried into that permafrost over time. And so a lot of it is organic rich. But it's very difficult to go to a soil profile and really see any differences with depth in terms of soil organic matter. One, that's because soil organic matter is very hard to analyze. And then 2, permafrost is also fairly heterogeneous. So there could just be variability there that's obscuring our ability to see any degradation of that soil organic matter. There was a study that came out recently showing an increase in acetate concentration with depth, suggesting that fermentation might be ongoing. But it's one of the few studies that have seen any kind of change like that with depth. And they might have just got the right soil profile to analyze. And they might have also taken samples that were old enough as well. Their oldest sample was 134,000 years old. So it took that long to kind of see any difference in that acetate concentration. 2 places where we don't detect microbial activity are Don Juan Pond, which is in Antarctica. This is a pond that remains liquid year-round. It's a little hard to see in this picture. The volume of the pond fluctuates by year, and it was low in this year. You can see a little bit of water from here. Most of it's back here. But this pond remains liquid year-round despite temperatures all the way down to -50 in the winter, because it is a brine. There's about 40% salt in this water, and a about 90% of that salt is calcium chloride. So that's how it can remain liquid at these super low temperatures. Of course, that means there's not a lot of water available for microorganisms in that brine. And there have been several studies attempting to document microbial activity. And while you can pull out some cells and some DNA, they probably just floated in there, got blown in, and are there and haven't completely degraded yet, but they are not active. So this is an environment where, yes, there's liquid, but it's not liquid. It doesn't have enough water to really be available to the microorganisms that are present. Another place where we have not been able to document microbial activity is in the University Valley soils in Antarctica. This is, University Valley is within the dry valleys. It's a slightly higher altitude valley, so it's much colder and drier than a lot of the dry valleys. The mean annual air temperature here is minus 23 degrees Celsius. So it does stay very cold all year round. And it's believed to have been about 150,000 years at about these temperatures and these conditions. It's very dry here. The soils that were sampled, and I'm talking about, are all from the valley floor here, not the walls. You can actually get nice organisms from the walls and see that they're growing in situ, but the floor is much drier and colder. It does get enough sunshine in the summer to raise the temperature of those soils a little bit. They come above minus 15 degrees Celsius for about 74 hours a year. And that number is important because it's above minus 15 where there's the potential for liquid water then to be present. But it's still not very much in this very dry environment, and for a very short amount of time, that is not enough for any kind of maintenance or repair even to occur and allow the organisms that got blown in there to survive. So primarily what they see here is, again, yes, you can isolate some DNA, you can see some cells, but it's very difficult to get any kind of viable organism out of here. And there's no activity. It's believed that there's really no activity happening in situ. So again, a place limited by the availability of water. All right. So from all this, we conclude that it's really the lack of liquid water at low temperatures that's the true limit on microbial activity. Interestingly enough, you can still get enzymes to be active at minus 90 if you put them in the right kind of liquid. So it's not really the low temperature so much as the water, but of course, those are intertwined because as temperature decreases, the availability of water decreases as well. Okay, I'm coming to the planetary protection here. All right, so it's important for us to understand how organisms on Earth can handle these frozen environments, because we are right now exploring frozen environments elsewhere in the solar system, and we're continuing to explore environments. We're doing it now, we're going to do it in the future as well, and we want to make sure that what we discover there, we can reliably interpret our data from those places, okay? So this is straight from the NASA's website on planetary protection, that the goals are to preserve our ability to study other worlds as they exist in their natural states. We can only do that if we avoid that biological contamination that may obscure our ability to find life elsewhere if it exists. And we have to ensure as well that we take prudent precautions to protect the Earth's biosphere in case life does exist elsewhere. Now, right now, we have robotic missions sent to Mars, but NASA is also tasked with sending humans to Mars in the next 20 or 30 years, so we'll see how that goes. And we are also currently exploring Europa and Enceladus. These are the 3 places in the solar system right now which we believe has the highest prospect for potentially having any kind of present or past life on them. Mars has a lot of ice. It's an ice-rich planet. Europa is a moon of Jupiter, has an ocean that is covered by a pretty thick ice layer. Enceladus is a moon of Saturn that again has an ocean and a fairly thick ice layer. Just in the last 2 years, we have a probe flying around Saturn, the Cassini probe, and it actually flew through some geysers that spew from Enceladus. So imagine a geyser spewing a little bit of water and ice from the surface of Enceladus out into space, and Cassini actually happened to fly through one of them and actually detected organics. So this is very interesting for the prospect of trying to understand life as we know it. Right now we only have one example, right? We'd like to know what that means in the big scheme of things. So these are really interesting places for study, and we want to make sure we can understand what does it mean that we find organics someplace. I'm going to talk a little more about Mars because I've done the most work with Mars and not so much with Europa and Enceladus. So we've had evidence for water on Mars for quite some time, but just in the last couple of years, we've had some really compelling evidence because we just have better, higher resolution instruments up there. And so here's just a few examples of where we see water on Mars. Most of it is in the near subsurface, and it's frozen. The atmosphere is very thin, so if ice is exposed to the atmosphere, it does tend to sublimate away very quickly. This is a recent impact crater where a meteoroid hit, threw up the material, revealed the ice there. The impact was probably about 6 months before that picture was taken. And then you see that, another 4 months later, we can't see much of that ice anymore because it's sublimated away. Those are in the mid-latitudes of Mars. The picture on the right there is from the Phoenix landing site. Phoenix is a lander that landed up in the northern part of Mars, closer to the northern polar cap. And it took its little scoop to shovel up some soil and examine it there, and revealed some ice right there, I think within like 4 centimeters of the surface of Mars there. Again, that sublimated away rather quickly, but showed us that it was there. More recently, we've got these, this is a cliff face right here, an exposed cliff. And we can see that all this blue is showing exposed subsurface ice sheets pretty close to the surface on Mars as well. That blue coloring has been added to highlight the ice sheets. I think it does look slightly a little bit blue, but it's certainly not that blue in reality. And these are in the mid-latitudes again, so we can see these on Mars as well. And this was pretty exciting for me, I thought, that we also just now have some evidence that there could be liquid water beneath the southern polar cap on Mars. And we see this on Earth as well. If you go to the Antarctic ice sheet and you go all the way underneath, there's liquid water under there, where the overburden of the ice keeps it liquid, and on Earth, the heat of the Earth is warming it up from underneath as well. Here, it's really important that that ice cap is on top, because it's preventing interaction with the atmosphere so that the water can't sublimate or evaporate away into the atmosphere. But very interesting prospects there. So there's a lot of frozen water here, and we'd like to know if that frozen water, 1, could be useful for humans when they go to Mars. 2, does it maybe contain some evidence of past life, or even just past organics might be interesting, or even some current life. And we certainly wouldn't want to introduce any Earth microorganisms in there, because that would potentially obscure our understanding of what has happened on Mars presently and in the past. So by combining everything we know about the limits for microbial life on Earth, their temperature limits, their water activity limits, that chart over there is limits of reproduction at different water activities, with the temperatures on Mars, and here's our temperature scale of both Earth and Mars. This is the average annual global temperature. This is the minimum and the maximum here for both of these. So you see there's some overlap. Okay, Mars is certainly much colder most of the time, but there is some overlap. And we want to take all that and define areas where we want to be extra careful on Mars when we go there. Okay, so we use all that information to define what is called by NASA special regions. And these are places where you want to just be extra careful, because you wouldn't want your Earthborne organism to get there and hang out for a while and obscure any future, your current mission or any future mission either. And so this is, the colors on this map show elevation, and the map then has been divided up into how special are the different regions on Mars. So Unit 1, which are the polar areas, there's a lot of near surface ice here. And so those are considered special regions because of the nearness of the ice to the surface, as we saw with the Phoenix lander probe there. And also Unit 2, there's a lot of ice as well. A lot of those cliffs showing ice sheets are from this area of Mars, the mid-latitudes. Okay. And then in the equatorial regions, it tends to get hotter and much drier. So there's hardly any ice there that we can really document. And if there is, it's certainly not very close to the surface. So we're not as concerned sending Earth missions there, because we don't expect much to be able to happen there either. Okay. So, in conclusion, I'm not going to be able to start my video here. Okay. All right, so we know that the permafrost microbiome is actively surviving those long-term low temperature conditions, and that helps us understand that there's a need to protect Mars from terrestrial organisms so that important discoveries can be made and correctly interpreted. And just recently, there's been a ton of great stuff coming out recently. They're like all studies published just this year. Organic matter has been detected on Mars in 3 billion-year-old mudstones in this crater. This is where the Curiosity rover is right now, doing lots of work. And we want to be able to correctly interpret that, right? If we didn't send a clean spacecraft to Mars, you would wonder if those organics just came off the rover, right? Are they really on Mars? You would want to know if we didn't send a clean spacecraft, could those organics have been altered by any organisms on that spacecraft before you analyze them? And so you really wouldn't be getting a clear picture of what these organics were on Mars, okay? And, you know, we do send clean spacecraft to these places, as clean as we can get them with our abilities, so that we can correctly interpret that data to try to better understand whether those organics are representative of perhaps abiotic synthesis on Mars, perhaps those organics got there from meteorites or comets that brought the organics there, maybe it's some evidence of fossil life. We don't know, okay? But of course, we have to be very careful with how we do those analyses so that we can better interpret what that means for Mars and then what it means for life in general. It would be nice to know if abiotic synthesis can happen on Mars too. That would be pretty cool. And as NASA has been charted with getting humans to Mars to do some more exploration as well, there's a lot of concerns for sending humans to Mars, and certainly just in space, because you know wherever you send humans, you send microbes. And my video here was a video of this astronaut making her dinner on the space station, International Space Station, and she has her tortilla and she puts her food on and then it floats away while she gets her next pouch ready of food, grabs her tortilla and puts it back on there, and then it floats away again while she puts her next pouch, gets her next pouch of food ready. And so, you know, we're bringing our own microbes, we're bringing the microbes associated with our food. They're staying in those space stations, in those space vehicles. And, you know, with all the awareness we now have about the microbiome and how important it is, NASA's just getting started on all these studies for how to understand the microbiome of the space station and space vehicles and how it impacts the health of the astronauts. And it's particularly if we're going to send them to Mars. How is their microbiome going to change with long-term confinement? It's going to take 2 years to get there and back. How is that, you know, if we put them on the surface, how's that going to impact Mars? Because their microbes are going to go with them. So lots of good things to think about if you have more of a human microbiome bent to your studies. NASA's doing a lot of cool stuff, and they're going to be doing a lot more in the future as well. So with that, I'd like to thank you for your attention. And of course, I don't do this work alone. Lots of people to acknowledge being involved in this work. Thank you.
So, the permafrost bacteria are taken as communities of microorganisms together, or is the total lack of diffusion essentially meaning that it's just you and your descendants in any given area?
That's a really good question. I don't think anybody's really done those detailed microscale analyses. I mean, it's just like most environments, you go and you get a gram of it, and you mix it all up and you see what's there. So, yeah, I don't think we know, but it's very likely that it is going to be your near neighbors, and that's who you're interacting with for the next 100,000 years. Yeah.
I was curious as to your thoughts of when you see the cold adaptation within the proteins and the signal was highest within the housekeeping genes. Mm-hmm. Why do you think you don't see more of a signal in the other genes that presumably are being used for proteins?
Yeah. I would think that it might just be that the core housekeeping genes have been around for longer in these organisms and being exposed to these conditions, and that those accessory genes are going back and forth more, and they might not have had the same kind of history of exposure to low conditions as the other ones. Again, it's not entirely clear. That could be it. That would be something to explore, yeah.
Well, I guess follow up on that, do you see any evidence of the flow?
I mean, you do, like you do in all organisms, some subset of the genes look like they were horizontally transferred, but I haven't delved into that in too much detail with these yet. Yeah.
Have people started to look at the zone where the permafrost becomes warmer from the heating of the earth underneath? And is there a different humidity there versus the higher?
That's a really good question. I don't think so, and I have a feeling it's just people aren't drilling deep enough. That it's challenging to drill through this, and I think they're just not able to drill as deep enough to get there. And where you can drill deep enough, like where the permafrost is shallower, it's probably also more patchy, the permafrost. So, you know, it's maybe shallower this way, but it also doesn't extend, you know, too far that way either. And so there's going to be other things going on. That's a really interesting question where you have those big continuous areas of permafrost with some significant depth. Yeah, I don't know. It'd be good to look at.
So I'll have questions about the implications for speciation when we have lunch. But I want to thank everybody. If you've got a white chair, will you take it out to the table? It's